This section of the digestive system chapter is going to look at enzymes in a little bit more detail. It's really building off of the functions that were just covered, but taking this side trip into the enzymes. This is a list of the functions we associate with the digestive organs. Obviously, the digestive system is going to digest things, and we'll look a little bit more closely at how that digestion takes place. The digestive tract is a series of organs that food moves through as we take the nutrient molecules out of the food that we eat, and so we have to move it through. Those organs are going to require secreting digestive juices into the tract, and we'll have to absorb the nutrient molecules out of it. And at the end, we eliminate the undigestible material that we have. Enzymes are part of chemical digestion. This is a table out of the book in the last module of the chapter, looking at all of the different enzymes. They've been arranged here based on where they're released along the digestive tract. However, I want to rearrange this information to look at enzymes based on the type of macromolecule that they break down. In the first part of this rearranged version of the table, I want to consider the enzymes that break down carbohydrates. The first two listed here are amylases, which break down starch, which is a type of polysaccharide. One's released in the saliva and one's released from the pancreas into the small intestine. And so we're breaking down starches in those two locations. The salivary amylase is important because we're interested in finding good sources of glucose in our food. And so if we can break down starches, we'll break it down into smaller molecules. It says here disaccharides and trisaccharides, but it is possible that some monosaccharides will be broken down as well. Those can bind to the glucose receptor on the tongue and will recognize the presence of a rich source of glucose in our food. We don't break down all of the polysaccharides that way, so there's more amylase released into the small intestine to finish breaking them down completely. Then the other enzymes that are listed here are all brush border enzymes, which means that they're present in the small intestine and they break down the small polysaccharides, the disaccharides and trisaccharides, into monosaccharides. I want to concentrate on the three at the bottom, maltase, sucrase, and lactase. They're each named for the particular disaccharide that they're responsible for breaking down. Maltase breaks down maltose, which is a disaccharide of glucose. So the product is going to be two glucose molecules. Sucrase breaks down sucrose, what we also call table sugar, which is a disaccharide of glucose and fructose, and so it's going to break it down into an equal mix of those two monosaccharides. And then lactase breaks down lactose, which is milk sugar, and that breaks the disaccharide of glucose and galactose down to those individual monosaccharides. I want to talk a little bit more about lactose. First off, let me point out that the monosaccharide galactose, which is specific to milk, is the milk protein, and lactose, the disaccharide, is just named as a truncated version of that monosaccharide name. Galactose hopefully reminds you of the word galaxy. We live in the Milky Way galaxy, and it's not a coincidence that the word for milk and the word for galaxy are linked together. A study that was published in the scientific journal Nature addresses the presence of dairy agriculture in humans' archaeological past and ties that to genetic presence of lactose tolerance. The hotspots that are highlighted here, the darker places in Northern Europe, Western Africa, and the Saudi Peninsula and Eastern India, all show places where there are genetic markers for people who are lactose tolerant. It also corresponds to data from the archaeological record of pot shards that were dug up and have molecular traces of dairy agriculture on them, suggesting that the production of lactase, which is not exactly common throughout the entire human race, is probably based on places where dairy agriculture took root. Now, what's not entirely clear from the data from this paper is that dairy agriculture dates back to about 8,500 years ago. And the presence of genes that allow for lactose tolerance probably only date back about 3,500 years or so. which suggests that there was quite a while for dairy agriculture before our genome was able to actually handle it. Most likely what happened is dairy agriculture relied on microorganisms to digest lactose so that adults could get nutrition from that. And that's where yogurt and those sorts of things came from. By using microorganisms to break down lactose in the dairy foods, it was possible to be digested by adult humans. These are enzymes that break down proteins. The first one, pepsin, is released from the gastric glands in the stomach, specifically the chief cells make it. And the others are released either from the pancreas or as brush border enzymes to work in the small intestine. Now, several of these have asterisks next to them because they're not actually secreted in their final form. For example, chief cells secrete what's called pepsinogen. And when it gets into the low pH environment of the stomach, it cuts off a regulatory section and becomes pepsin to break down proteins. This is necessary so that we don't digest the proteins in our cells before that enzyme's needed in the proper place. Carboxypeptidase, chymotrypsin, elastase, and trypsin all work on similar processes, but not pH, because the pH is neutral in the small intestine, but something else will actually make prohormones of those get altered to the active hormone form. Enzymes that break down lipids are called lipases. There are 3 different types of lipase. There's lingual lipase, which is secreted from glands in the oral cavity, gastric lipase, which is secreted into the stomach by chief cells, and pancreatic lipase, which is secreted from the pancreas into the small intestine. Lingual lipase is actually more active in the stomach because it works best at low pHs, along with gastric lipase. And then pancreatic lipase is going to be active in the small intestine at a neutral pH. Lipases need to be emulsified, and so the emulsification that's capable to break down lipase in the stomach is more of the mechanical emulsification as the food's being churned around, kind of like if you shook oil and vinegar salad dressing. But once lipase gets into the small intestine, emulsification is handled by bile that's secreted at the same place where the pancreatic enzymes get into the small intestine. When we think of nutrient molecules, we usually think of carbohydrates, proteins, and fats, or lipids. We don't consider nucleic acids as nutrient molecules. We don't see them on a nutrition label on our food. However, they are present in our food, just not at levels high enough to be nutritionally relevant. The enzymes that break down nucleic acids are called nucleases. There's a type of nuclease released from the pancreas, and enzymes that break down smaller nucleic acid fragments at the brush border. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. Enzymes are named having an ASE ending, and then the rest of the word tells you what it breaks down. Amylase breaks down starch because amyl is referring to starch. Nucleases break down nucleic acids. Lipases break down lipids. Some enzymes have special names like pepsin that are a little bit old-fashioned and therefore don't use the ASE ending approach. But pepsin is referring to a peptic enzyme, meaning that it's in the stomach.